much. Thank you, everyone. It is lovely to have the opportunity to speak today. I have to say, I was telling Bert that I don't love the virtual presentation anymore, and I feel like I lose the ability to sort of converse with the audience. And so I have my chat up, and anybody who, anybody who wants to sort of pipe in and ask questions along the way, please feel free to do so. And can I ask, did I just switch the displays, or do you see the display appropriately?
It's appropriate. Yeah.
It's appropriate? Okay.
Yeah.
Okay. So I am going to start by first acknowledging the people in the lab who did the bulk of the work. Many of these folks you will have seen around the school. A lot of the work was done by a really talented postdoc, Chin-Yin Liu, who's about to start his own lab at the University of North Carolina. And I will highlight the work of these other people as we go along. As many of you know, my lab has had a long-standing interest in this question of what drives the variability in terms of TB and infection and treatment outcomes. I think the global numbers really are so interesting in terms of thinking about the variability in outcomes when we say a third of the world or some version of a large number of people in the world are latently infected, but only a subset of those people are going to go on to have active TB, either primary progressive TB or reactivation TB. It is very clear sort of the problem that we're dealing with in terms of the variability in infection outcomes. And I think to some extent the same might be is true in terms of thinking about treatment outcomes, in terms of thinking about treatment outcomes. So even in the setting of TB treatment trials with a conventional first-line treatment, 5 to 10% of those individuals will fail treatment. We have 450,000 cases of MDR TB every year, despite the sort of very steady drumbeat of DOT since the 1990s, which was supposed to constrain the emergence of new drug-resistant strains and limit the spread of drug resistance. And so it just highlights the fact that there are determinants that we don't have an understanding of that will continue to plague us as we try to limit this epidemic unless we get a better handle on them. Now, this is especially important in terms of thinking about treatment outcomes, and everything I'm going to talk to you about today is in terms of thinking about bacterial variability as a determinant of treatment response. Because we are in the golden age of new treatment. Well, golden age might be a little overstating it, but sort of clearly reaping the rewards of 30 years of drug discovery efforts with new drugs for TB that have been rolled out, both bedaquiline and bertomimid, as well as repurposed drugs like linezolid that have made new all-oral regimens for MDR TB possible, and thinking about shorter course regimens for drug-susceptible TB also possible. And so there has been enormous progress, and there are a number of trials underway that further promise to push the needle. This is in the context of what I'm going to call the programmatic framework of thinking about TB, which is a one-size-fits-all model. As you know, the TB is treated in a resource-limited framework, and the pressure is for a simple one-size-fits-all regimen. And inherent in this is the idea that if new drugs come out to which the organism has not been exposed previously, that we should expect no pre-existing resistance to those drugs, and that we can roll out these new regimens in the absence of diagnostics for drug resistance, because if applied appropriately, So if these new drugs are applied in combination together and there's no pre-existing drug resistance, there's an assumption that, at least a hope that there will not be the emergence of drug resistance. And so we're going to come back to that as we move along. We're going to talk about drug resistance in the both conventional and sort of more nuanced sense of altered antibiotic susceptibility today. And in order to frame that, I wanted just to remind you what the conventional framework for thinking about TB drug responses is, and that is a binary framework of resistance and susceptibility, whereby bacteria are plated on a critical concentration of drug, and either you have growth or no growth, and that is dichotomous. So that drug resistance is dichotomized into resistance and susceptibility, and has been the basis for an enormous amount of genomic effort to understand the genetic basis of this high-level drug resistance. And this has led to a pretty good understanding for the first-line drugs of the genetic determinants of high-level resistance, and has enabled the development of nucleic acid-based diagnostics for drug resistance. Now, against this backdrop, an understanding of resistance, let's see, it is clear that there are more nuances in the bacterial response to drug than this dichotomized understanding of drug resistance would suggest. So that if you measure the MICs to the first and second line drugs, you'll see clearly a distribution of MICs. I'm showing you RIF here, and I'm not sure you can see my pointer, but so I'm showing you RIF on the left, and you can see.
Yeah, we see your pointer.
Oh, okay. So you can see a bimodal distribution of RIF MICs, and here is the current the breakpoint that would discriminate resistant from susceptible strains. And you can see even within susceptible strains, there's a distribution of MICs, and clearly for something like ofloxacin, the distribution is much less clearly bimodal, and you can see the indicated breakpoint, but that there is not such an obvious distinction between resistant and susceptible strains. And in as much as it has been studied, which I will say is not very extensively, it is also clear that even within strains that would be considered susceptible by the dichotomized MIC measures, that the MIC appears to matter for treatment outcome. So here we're looking at work from David Lahn's lab, where he's looking at distributions of MIC to rifampin among drug susceptible strains, and that there is an increase in the risk of relapse if you are closer to the MIC, the breakpoint MIC, than if you are a more susceptible strain. So even drug susceptible strains have differences in drug responses that are likely contributors to treatment outcome. And this dichotomized version of MIC is a very simplistic view of drug responses, because in the laboratory settings, it's become clear that in addition to resistance, which is if you're plated on drug, the bacteria can grow on drug. There are different forms of altered antibiotic susceptibility. The best two understood of which are antibiotic tolerance, which is if you're plated on drug, you may not be able to grow, but you're not killed. And antibiotic persistence, which is a form of antibiotic tolerance in which there is a highly drug tolerant subpopulation that is able to persist for an enormously long period of time in the face of high levels of drug. So those have been speculated to be important in terms of thinking about TB treatment responses, but clinically have not been well defined. So our lab had been interested in thinking about determinants of TB treatment responses, and we had hypothesized that TB has really been under human TB. It's an obligate human pathogen. It's been under drug pressure essentially for 80 years. And that under the sort of tip of the iceberg of clinical resistance, there might be a whole tapestry of altered drug responses that might affect responses to the new drugs, as well as responses to the existing first and second line drugs. And so we have spent quite a bit of time over the last 5 years using population genomic approaches to begin to elucidate those. And we have, and our lab and work of David Lon's lab, Chris Sessetti's lab, other labs in the field have found evidence for mutations in the bacterial population that cause drug tolerance or sub-MIC resistance that are associated with certainly the acquisition of canonical drug resistance and potentially the acquisition of relapse in drug-sensitive patients. And so what I want to tell you about today is actually an effort to really expand that understanding, starting with an evolutionary perspective. And this is taking advantage of the fact that, and sort of the really disturbing observation that that where TB has typically been considered a relatively monomorphic, genetically monomorphic pathogen, there's more genetic plasticity in the organism, even in the context of a single infection, than previously appreciated. And so this is a study actually from Xinyun Liu when he was a postdoc in Chen Gao's lab in Shanghai, where they were looking at patients on first and then second line drugs, looking at the bacterial populations in their sputa using deep sequencing. And what I want you to take away from this is, here are the first and second line drugs. This patient started with a diversity of mutations, started with a diversity of mutations, but rapidly acquired quinolone resistance mutations, even though they're on a multi-drug regimen. And you could see, tracking the resistance mutations via deep sequencing, you could see the emergence of and then subsequent fixation of those quinolone resistance mutations. Those are in GYR-A, and then the subsequent emergence of mutations that establish resistance to isoniazid, pyrazinamide, and a compensatory mutation for rifampin. And so we wanted to leverage that approach and look more broadly at the sorts of mutations that were arising in patients over the course in patients, to understand the selective pressures that the bacterium faces in human populations. And so we took advantage of the fact that when a TB sputum sample is sequenced, the whole sputum sample is sequenced. So the standard in the field is not to do single colony purification, but just to sequence that whole sputum sample. And so you can see in that sputum population both fixed mutations, so mutations that distinguish the population broadly from other strains, other TB strains, but you can also see unfixed mutations that represent potential targets of ongoing selection in that patient. And so we applied that approach to actually all of the whole genome sequencing data that was publicly available at the time. So it was, we took all 51,000 TB genomes from NCBI, in the absence of metadata, reasoning that some of it was going to represent patients at different points in times of treatment, and others were going to represent, there's going to be a diversity of kinds of sequencing in there, but we were going to look for unfixed mutations as potential targets for ongoing selection within individual patients. And then just some inside baseball, for every strain, we use the cryptic database to genotypically define DSTs. So then we took all of those unfixed mutations, and I'll just go back, let's see. Oh, it's going to come up in a minute. And then we applied a number of statistical tests looking for selection. So we are going to look for both mutational burden and a D&DS ratio as a measure of selection. And this is what the data look like. So here I'm showing you the mutational burden along the y-axis. So this is the normalized number of unfixed mutational events. These are genes. So these are the most highly variable genes lined up along the y-axis or along the x-axis. And I have indicated with black diamonds the genes that are actually known drug resistance or drug tolerance genes. And what you can see is that all of these drug resistance genes for first and second line drugs are sort of at the top of the list. But you can also see that there are a number of genes that are mutating as much as known drug resistance targets that we would not have otherwise a priori linked to drug resistance. You can also see that there's a very strong non-synonymous to D&DS ratio in these genes, again, consistent with very strong evidence of positive selection for these genes. And you can we also scored intergenic regions, so you don't have a D&DS ratio here, but you can also see a number of intergenic regions that have a disproportionately high number of unfixed mutations. And so to understand what is driving this sort of positive selection outside of the known drug resistance genes, we just started with a gene at the top of the list that we didn't know anything about. And that gene is RV1830. We're going to rename that gene in the next slide, ResR, and that's going to become obvious to you in a minute. And we just started this as a way to begin to understand selection broadly across TB genomes. So here, ResR is an essential transcription factor. Actually, many of the genes that are under selection are transcription factors. The mutations are lining up in the predicted DNA binding domain of that gene, of that transcription factor, suggesting that they're causing altered regulatory activity. And the evidence of selection, you can see mutations in both drug susceptible and drug resistant strains, but the signal of selection as measured by the D&DS ratio is higher in drug resistant strains, suggesting that it could be associated with antibiotic selection, but it was unlikely to be causing resistance to a first or second line drug. And so we wanted to understand what the phenotype was, and we're a basic science lab, so we're our first tool of choice is genetics. And so to understand what is driving that sort of phenotype in the TB, in clinical TB strains, we used a tool called oligorecombinating to create isogenic allelic variants in ResR that reflect the clinically common mutations. So we reconstructed 3 different point mutants. We constructed 2 parallel mutations of each. We compared them to strains that had been recombinated, but recombinating is not perfectly efficient. So you get strains out in which you did not get the the SNP recombinant in, and those are the control strains. And we confirmed all the strain identities by whole genome sequencing to make sure that we hadn't acquired second site mutations. And then we just started phenotyping to try to understand what these variants were doing. The first obvious phenotypes to assess, because we had this sense of an association with drug resistance, was whether they caused either drug resistance or drug tolerance. And we tested resistance and tolerance in the face of 8 first and second line drugs. They did, the Resar variants did not cause significant changes to the MIC of any drug except isoniazid, which there was a small increase in MIC to isoniazid, which we could be clinically relevant, but I'm going to show you a much stronger phenotype later that I think is the clinical driver. But there is a slight shift, increase MIC to isoniazid, but by and large, the resistance and tolerance phenotypes are not very striking. But when Chinyin was doing this, he had this totally amazing phenotype. So he was watching his plates, and here I'm showing you, so this is the tolerance assay. So you take TB, you expose it for 24 hours to 100x the MIC of a drug, and here I'm showing you isoniazid or rifampin, or no drug is the negative control. You wash that off and you plate. And he was watching his plates, and he noticed that in response to drug, there's a delay in growth. So here is no drug. Everybody starts growing sort of almost by about day 12. In response to drug, that's delayed, but that delay is significantly abrogated in the Resar variants after treatment with rifampin or isoniazid. So it was not drugs specific. And indeed, it was, you saw the same phenotype. You saw the same phenotype after treatment with drug combinations. So here are the various combinations of the first line drugs. You can again see that the Resar variants cause this very interesting sort of faster recovery phenotype. And we could quantitate that. So we developed Junhao Zhu and Eric's lab developed a colony tracking software that enabled him to quantitate this. And so here you can see that regrowth phenotype for the Resar variants and wild type strains quantitated, and that allowed us to, we quantitated it for the 8 first and second line drugs. And what became clear was that antibiotic treatment causes a delay in colony formation by the surviving cells, and the Resar variants reduce that delay by between 25 and 50%, depending on the drug. Okay, so that's sort of at this very macro level. We could see a similar phenotype if we looked at a single cell level. So here I'm showing you TB that we have labeled, used a fluorescently labeled D-amino acid to label the cell wall. And then you can change the color on the fluorescently labeled D-amino acid to do a pulse chase labeling and track new cell wall biosynthesis. And so in these experiments, we label the cells, we do a 100x MIC antibiotic challenge, wash out the antibiotic, and then chase with a different, like a blue floor, and look at cell wall regrowth. And it's just a very nice way of showing you what I showed you on at the colony level, that after drug treatment, the Resar variants recover, start synthesizing cell wall significantly faster than wild type cells. And we can quantitate that by flow cytometry, and you can see for every drug we tested to differing degrees, the Resar variants are going to recover faster. So we're going to call that resilience, and that's how we got this name Resar for RV1830, which is the gene name for TB. And then it's a transcription factor, right? So we wanted to understand what it was regulating. And so we use this method called ADAPT-seq, which is basically like in vitro ChIP-seq. So you take a purified protein, incubate it with sheared genomic DNA, and then look for the bound DNA fragments that you enumerate by sequencing. And when we did this, we found 4 high affinity ResR binding sites, one of which we had seen before, because it was actually an intergenic region that is actually one of the highest targets of selection in the TB genome. So this, and then we could show you that that ResR binding site, if you map all of the genomic variants, the binding site itself is what is accumulating mutations across clinical strains of TB. And we could show that that ResR is actually a positive transcriptional regulator of this gene WEB2. And these variants that are accumulating in clinical strains, when we again created isogenic allelic variants in TB, phenocopy the ResR mutants, suggesting that this is actually selection for the same phenotype. And what we're mapping by tracking the signature of selection is we're mapping a new antibiotic survival pathway. And I'm going to just cut to the chase and show you how this turned out. So we had ResR, which is a positive regulator of WEB2, and we have positive selection on diversification of the WEB2 promoter where ResR binds. WEB2 is actually also under positive selection, and WEB2 acts with the transcriptional cofactor WEA. It is also under positive selection. This WEB2 binding site is actually contains a perfect palindromic repeat that is present in one other place in the genome, right near the origin of replication. It is also under diversifying selection in TB strains. So here it is, one of the top hits in TB, diversifying selection. ResR actually binds here, albeit at lower affinity than it binds to this WEB2 FBIA binding site. And it also binds DNAA. DNAA also binds in this binding site, and DNAA is also under positive selection. And we had previously looked at DNAA as a mediator of drug of sub-MIC resistance to isoniazid, because it has that same slight shift in isoniazid phenotype that we had seen for the ResR variant before we recognized this regrowth phenotype. But now we understand that actually what we're looking at is a macro pathway that is regulating core cell growth and division processes that somehow leads to this faster regrowth phenotype after drug, but that's highly selected in clinical strains. And it is interesting to think why we are seeing this phenotype in clinical strains. This, what I called growth delay, is actually pretty well recognized in the drug development world. And it is called the post-antibiotic effect. So this is a figure from a paper from 1969, where they're showing the post-antibiotic effect for isoniazid and TB. And post-antibiotic effect has been recognized for TB and other pathogens for over 50 years. The mechanism of the post-antibiotic effect is unclear, but the post-antibiotic effect is an important feature in thinking about how dosing regimens are developed. So there is an assumption that a long post-antibiotic effect will allow antibiotics to be dosed less frequently. And as you might know, in TB, one of the programmatic constraints on for TB is actually the assumption that TB treatment will be more effective and there'll be less resistance if patients receive directly observed therapy. But directly observed therapy has been programmatically difficult to implement. And so while you can have programs that administer directly observed therapy daily or 5 times a week, there are also approved DOT regimens for administering drugs 3 and even 2 times a week, which, at least to my mind, raises the question of whether in developing intermittent dosing to facilitate DOT, we might have selected for a new class of antibiotic resistance, which are what I'm going to call the temporal escape mutants. Okay. And the other possibility, I guess I would say, the other possibility is that this class of variants is selected because antibiotics and antibiotic selection and host selection might actually favor the same kinds of survival pathways, so that there is a body of literature that suggests that antibiotics impose oxidative stress on the bacterium. It is important to note that both RESR and WEB2 are redox sensitive regulators. And so it is also possible that antibiotics and host derived oxidative stress are driving the same sorts of adaptive escape mechanisms. Again, selecting for variants in core cell growth and division processes. However those are being selected, what is clear is that these resilience pathways variants are actually kind of shockingly common. So between 2 and 10% of drug susceptible strains in high burden countries have a resilience pathway mutation, and the resilience pathway mutations are even higher in drug resistant strains. And there is some evidence to suggest that the resilience pathway variants are also associated with relapse after treatment for drug susceptible TB. So there are not a lot of well-curated treatment data sets, but we took advantage of the REMOX TB trial. So this was a global phase 3 clinical trial, which sought to reduce TB treatment duration for drug susceptible TB from 6 months to 4 months using a moxifloxacin containing regimen. In this regimen, or in this trial, which actually failed to, the regimen failed to shorten, or was associated with worse outcomes. There were 36 recurrent TB cases. And what you could see is that the relapse in the REMOX trial, so even in a trial setting, relapse was associated with a higher prevalence of resilience pathway mutations, and that in some patients, what you could see is that the resilience pathway variants either emerged de novo and were fixed, or were pre-existing and then became either more highly prevalent over the course of treatment, or fixed during the course of treatment, consistent with the model that they provided a selective advantage. So we are very fond of population genomics as a way of identifying bacterial variants that are important in shaping treatment outcome. But we had wanted to explore other methods for finding important bacterial drivers of treatment outcome. in part because there's some limitations of using population genomics in TB. The most important of which is that TB has a very high clonal population structure, so it has no recombination and no horizontal gene transfer, which creates a problem of genetic linkage and which limits the power of GWAS studies. This is the phylogenetic structure of TB. And the other thing you will note is that it is highly geographically structured. And so there are lineage and sublineage associated there is lineage and sublineage associated variation in the epidemiology of TB and association with both virulence traits and treatment outcomes, but those are obviously, of course, confounded by this geographic structure. And so while a bunch of studies have suggested bacterial determinants of both infection and treatment outcomes, in our minds, the biologic plausibility of these associations is always unclear given these both geographic and epidemiologic compounding factors. And so we had wondered whether or not we could come up with a new new method for looking for biologic drivers. And I put this slide in kind of in a weird way, I realize, but with a special sense of urgency, given the new drugs that are coming out, wondering whether there are drivers that are going to affect both treatment response to the existing antibiotics, but also to new drugs as they are being rolled out, recognizing that the new drugs, and especially bedaquiline and protomanid, have really whole new classes of antibiotic targets, which might be under host selection in different contexts. So we were interested in generating a method for defining intermediate phenotypes of TB in a way conceptually similar to approaches that have been used in human genetics, such that in order to improve our ability to find bacterial phenotypes by associated with treatment outcomes, by finding bacterial intermediate phenotypes, linking those to specific genes, and then using that as a bridge to do the genotype to phenotype associations. The approach that I'm going to show you, we prototyped in a cohort of 1,600 TB patients from Ho Chi Minh City, Vietnam. It was actually a population dynamics study, but they followed these patients, and 16% of the drug susceptible patients with drug susceptible TB had delayed sputum clearance, after which they defined as persistent smear positivity after 2 months of treatment, and 42% of the patients with drug-resistant strains had persistent smear positivity. But beyond the association with drug resistance, this cohort was underpowered to do a GWAS versus treatment outcome because of the genetic linkage problems. And so we decided to take an intermediate trait and use these strains to develop an intermediate trait analysis platform. And we down selected from the 1,600 strains, about 160 strains for intermediate trait analysis. The intermediate trait analysis platform was developed by Sydney Stanley, who is a graduating PhD student in the lab, and it was an unbelievable amount of work, but it is so beautiful. So she actually genetically barcoded these 158 TB strains in biologic triplicate such that they could be pooled together and profiled through various competition or various growth conditions in basically a pooled competition assay where the function the fitness phenotype of every strain could then be elucidated by deep sequencing. So she created the barcoded pool, profiled it through 8 antibiotics and whatever, 7 different carbon sources. And out of those, enumerated the fitness phenotype of every strain under each of those conditions. And I'm just going to show you this data at a very high level because I'm going to use it just to make a very simple point. First, so this is all 160 strains, and here are the fitness phenotypes under all the carbon and drug conditions. And you can see there is phylogenetically structured variation in the phenotypes to both carbon and drug, carbon source and drug. And you can see that there is one notable outlier subclade, which is called lineage 1.1.1.1, which is a metabolic outlier. So it's slow growing under nearly every in vitro condition, but it is intrinsically more resistant to protomanid, even though it has never seen protomanid before, right? So these strains have not been exposed to protomanid, but this subclade is intrinsically more resistant to protomanid. If we do bacterial GWAS against these traits, we can find genetic associations with many of the traits, but the strongest one is the association that links the 79 SNPs that define this lineage 1 to the 4th, I guess we'll call it, with its associated growth phenotypes. You will see that the strongest associations we find in general are with slow growth phenotypes. All of these, the mutants are growing slower than wild type, except this one gene, which is associated with improved fitness in rifampin. But in general, what we're seeing is selection for slow growth. But even though things look slow in vitro, if we then link those, the genes that came out of the intermediate trait analysis with the clinical traits, what you can see is that we find both lineage 1.1.1.1 strains and mutations in RV1339 are associated with bad clinical outcomes. So this sublineage is actually associated with both cavitary disease and treatment failure in Vietnam, and the RV1339 mutants are associated with treatment failure. This lineage 1.1.1.1 is also associated with higher apparent transmission if you look in a branch length analysis, suggesting that actually slow growth in vitro might mark a metabolic state that is generally advantageous to the bacterium. For those of you who are sort of bacterial phylogeny aficionados, I'm just going to use this as a moment to say, actually, they're really cool and sort of unexplained structure in the distribution of TB strains around the world. And this sublineage in particular is only found in Vietnam and Thailand. And the Vietnam and Thai subclades of this lineage are very independent of one another, even though the strains are about 200 years old, and there's been a lot of population movement, especially from Vietnam during the Vietnam War. But there is a lot of more geographic restriction of this subclade than one would have otherwise expected. And then I'm just going to cap this discussion of metabolic selection with a very brief case of actually a strain that we have been studying in collaboration with Megan and Shoma Radachuri. And I'm not actually going to go into the whole back history of this this cohort, but just want to highlight this strain, which it came out of a genome to genome association study, which I'm not going to go into. But just to say that this is a strain that is actually locally very successful in Lima, Peru, that is not associated with drug resistance, and not explained by epidemiologic drivers, but is as a transmission success story in Peru. And as we dove into the sort of biologic differences in this strain that might explain its transmission success, what we found is that it actually had altered redox homeostasis. And I don't want to dive too much into redox balance in TB, except to say that it has a gain of function mutation in a key redox active protein that shifts its redox balance to an oxidized state and may be generating success in the population because it leads to reductive stress resistance. And the thing you should know about reductive stress resistance in people is that the bacterium is under reductive stress in hypoxia, should sound familiar because of TB granuloma and low pH. And so there are host metabolic conditions that might be leading to this altered redox balance in the bacterium. But the reason I bring it up today is actually this, that it is also more resistant to protomanid, even though these strains have never seen protomanid, which should sound very familiar to those Vietnam strains. Suggesting that there is host selection for redox variants that might be altering susceptibility to the new energy targeting antibiotics that could lead to a more nuanced landscape of antibiotic susceptibility in clinical TB strains than our current drug rollout strategies are allowing for. So, in summary, we would say that TB, like every pathogen, but although perhaps not like the TB community might have imagined, like every pathogen, TB is constantly evolving. The evolutionary solutions that TB finds might not be what you would normally expect from a host pathogen-driven evolution. So we are not seeing evolution of, say, immunologic targets, but we see enormous pressure to evolve bacterial metabolism and cell growth characteristics. And we think that we will need to pay attention to this because the new TB drugs target energy metabolism, and there may already be extensive variation in susceptibility to those drugs driven by host selection. And now I just want to put in a last plug for TB drug resistance testing. The sort of one-size-fits-all regimens for TB have been they've been sold on the basis that we won't need to do drug resistance testing anymore because new drugs mean no resistance, and then no combination therapy means resistance won't emerge, and so we'll be home free. Same pills for everyone, no resistance testing needed. And I think that that is just incredibly biologically naive. But that there is a moment available to us now to leverage especially the sequencing capacity that has been built out for pandemic and COVID surveillance, to leverage that for both AMR surveillance and specifically resistance testing and TB. And I would like to advocate for what I call the personalized public health approach to TB, such that we get out of this sense that TB has to be treated in a one-size-fits-all model. And I noted the people who did a lot of this work